Understanding diabetes and obesity. Fruit flies as model organisms. Obesity caused by an inability to properly use insulin, corresponding type 2 diabetes, is a serious issue facing public health in the 21st century. Over 20.1 million people in the US alone have some form of diabetes, 90% of which is type 2. Type 2 results from the inadequate use of insulin, usually caused by diet, lifestyle choices, and other genetic factors. But what does insulin do? Insulin is absolutely critical in metabolism, which is how humans get their energy through a sugar molecule called glucose, which is derived from our diets.
That looks delicious. May I have some?
You're right, Fred the fruit fly. We're in the middle of a video right now. You can't eat.
Buggers. That's a load of rubbish.
After we eat, glucose is absorbed into the blood through the intestines. As a response, specialized cells in the pancreas release insulin into the bloodstream. Cells in the body can then absorb glucose, using it for energy or converting it into. Obesity is a known risk factor for type 2 diabetes, and they often go hand in hand with one another. For example, it is known that diets high in fat tend to have a relationship to insulin insensitivity that is characteristic of type 2 diabetes. The relationship between the two is an area of active research. For example, understanding how insulin signaling is regulated is absolutely essential to fully understanding the nature of the two diseases. Could it be genetic? Could it be due to diet? Or could it be a mixture of the two? Research suggests that there may be a role for certain building blocks of proteins called amino acids in the regulation of insulin production in humans. For example, significantly increased levels of the amino acids tyrosine and phenylalanine have been found in individuals who are type 2 diabetic and obese.
But wait.
Tyler, what is the problem?
Well, Fred, how can these two molecules influence something as complex and important as insulin signaling in humans? As you can see, there's a spiderweb of metabolic pathways for both phenylalanine and tyrosine, meaning it can have multiple different products and multiple different effects.
How can we actually know?
Well, I'll tell you, Fred. A study on the worm C. elegans suggests that tyrosine can affect insulin signaling by acting through a complex pathway on a regulatory gene called FOXO.
Whoa, that looks convoluted in nature.
With this in mind, we can further deduce the role of tyrosine and phenylalanine in insulin regulation by using genetics. Fruit flies make excellent model organisms for this because of their easy maintenance and the fact that they're easy to genetically manipulate.
We do? Well, that is quite...
Researchers at Marquette University are asking the question of how tyrosine has a role in the egg production, also called oogenesis, of fruit flies. The basis for this research is the fact that flies that have a mutant gene called TDC1 show severely reduced oogenesis, likely due to elevated tyrosine levels. Because when you shut down this TDC1 gene, shown in the figure here, tyrosine has nowhere else to go and thus accumulates within the fruit fly. Could a pathway linking elevated tyrosine levels to decreased egg production be working through insulin signaling, also through the regulatory gene called FOXO, as was discovered in C. elegans? Well, this remains to be seen, and will require an intensive look at the hypothetical pathways shown here. The more we know about the genetic and molecular basis behind insulin signaling, the better diabetes and obesity can be understood.
On behalf of all of the fruit flies in the Queen's realm, we agree.
Thanks for watching and have a great day.